Why? I'm good. I'm trying to broadcast this. Yeah. Okay, can someone go to my Facebook page and see if this is streaming to you guys? Thank you, sirs. Yeah.
Not working on my phone.
Okay, that's fine. Oh, okay. Wi-Fi. Oh, it's mirrored.
Yeah.
It's okay if you can't. It's all right. Ew.
It's a good thing that you came prepared.
Why?
The cookie company called and said that they weren't going to be able to deliver until 2 o'clock because of icy streets.
Oh.
So I told them to take the cookies and
Take it off.
put them somewhere.
Yeah.
Well, at 2 o'clock, they're not going to do me a lot of good.
All right. Why does this look like that?
It looks like shit.
It does.
Okay. I don't know, somebody's head's right in the way.
I'm trying to system on.
We need it's got to be the resolution.
Resolution. Okay.
So why is it going?
Did you go up to screens?
But yeah,
Here,
why are you
here.
why is it
I don't
coming out
know.
here?
It got the from this guy. And this is a different one.
Because I know I have mine hooked to another monitor too, and sometimes you have to go in and like change the
Okay.
size of the screen.
Sure. Oh, I know where this is. It's the settings, right?
Yeah, system preferences. And this is what I'm getting at.
That's perfect. That's perfect. That's what I want to show you. Thank you. Displays. Is that right?
Yeah.
Okay.
I don't know if to try one of those.
Try display.
Or try a size.
Yeah.
Doesn't look good.
Is that okay?
There we go.
Better? Okay. Thank you. Oh, I have cookies, Eric. Do you have a PowerPoint, Eric?
Do I have a PowerPoint?
You don't? Okay.
I don't have any. Off the cuff.
You introduced this.
I did. I really need to do
Yeah, it works. All right.
Hi, Amy.
Amy.
Hello. She won't be there.
Okay.
Hi,
Tom.
Do you want to check the stream?
Okay, so they
It's fine.
It's okay. It's gonna work.
Okay, then, as long as this is fine, people still eat them. Thank you so very much for doing that. We are no longer cookie lovers here.
Trader Joe's cookies.
I guess what I told them is stick to cookies. Maybe they will speed things up.
Yeah. So I still have time to go eat.
Yes, I know, that's what I said. Oh. There's often been a disconnect.
Yes, I am aware. I didn't like nails. Yeah, I don't understand. But what's it? Yes, and today it was a chip. Yeah.
It's okay.
Thanks. Oh yeah.
Okay, why don't we get started? It is my distinct pleasure to welcome our speaker today, Han Lee, for his thesis defense talk. I also thank all of you for braving the elements and for braving the fact that we have competing thesis seminars going on at the same time. So the fact that you lobbied in on this is greatly appreciated, not only by myself, but when I was sitting and thinking,
Something is wrong with this thing.
so maybe we won't have.
No! But it doesn't
a
look.
roller coaster.
Oh, good boy.
It's animated.
All right, before I change the display again. Sir,
Don't mirror displays.
don't mirror displays. Okay.
Photo arrangement.
Okay. Ah. Is that okay?
And then change the resolution.
Whoa. Display. Back.
Down this window.
Hmm? Oh shit.
You now actually have two screens. Go back to where you were. Go over to this screen.
Oh.
Mike, would you like to come?
Yeah, yeah.
He has a PhD.
Yeah. Ouch.
And it should be easy, you think.
The resolution is in display, isn't it?
Oh.
There it is.
Oh, that's right.
Oh.
Okay, that's fine.
Let's see. We want probably this.
Is it better? Okay. Okay.
And then if you just do presenter tools.
Yeah. I can't find my mouse now. Oh, it's there. Okay. Okay, so I think it works now. Okay, sorry, thank you.
We're good to go.
We're good to
I was going to start my comments by noting the fact that Han has been a graduate student for 6.5 years, but maybe now sort of some people kind of shit the feeling here. But so I was looking at this from the very beginning, and it actually has been a long time, but I think it's all turning out for the very best. At least, I certainly believe so. So I looked through my notes, and Han and I met for the very first time, I think back in September of 2007. My very first note of him sitting in my office was from October of 2007. And at that point, he joined my lab and started working on a number of CNHEJ genes, classic non-homologous end-joining, mostly in conjunction with a senior graduate student, Farzana Fasid. And for about the next year and a half or 2 years, actually things went along extremely swimmingly. They were very productive together. They ended up publishing 2 papers together. Farzana's first author on them, and Han is a junior author, I think a third or fourth on the one, but he's second author on the major paper. And that actually turned out to be a super paper. That paper came out in like 2010 or something, and it's already been cited over 100 times. And much of the repair analysis in that paper was due to Han's work. So I thought a very fine start to this graduate career. This was followed by what we sort of refer to as the 3 dark years sort of thing, where after this lovely sort of opening trajectory, we decided that one of the things we're going to do is move our targeting studies into embryonic stem cells. It was at the same time that my laboratory was semi-imploding. I was having kind of a funding crisis, and the solution appeared in the person of Mary Burpo, who was doing gene targeting over at the Stem Cell Institute, was doing exactly the project we wanted to do. So for a year, Han physically went over there, and that turned out to be a disaster from all sorts of perspectives. The project just did not work. There were some personality questions there as well. And I think it was to everybody's great relief that after a year in purgatory in Siberia, not so cheap, but in Siberia, that he came back to my lab. Okay, so there was sort of a year that was just sort of disappeared. Right on. And then after that, things didn't really improve a whole lot for the next year and a half or 2 years. So we have these beautiful projects. We weren't going to do it in stem cells anymore, but we were going to do it with zinc fingers, and we're going to look at the impact of DNA PK on all of this sort of thing. And once again, for a whole variety of technical, theoretical, biological reasons, those projects simply did not pan out. And it was not for lack of trying by anything. And so we got to about 2011. I have here June of 2011. My personnel's going like, shit, okay, none of this is working. We need something new. And it was in that meeting that I sort of said, Han, how about tackling nucleases? They're so friggin' important. We really have no idea what they're doing, how they work in these cells, and they seem to control everything. Would you like to investigate them? And much to his credit, he sort of said, sure. I think at that point, after 2 or 3 years of really just one sort of bad thing happening to him after another, a lot of people might have just tacked it in and gone home. But Yuhan has always had an enormous amount of enthusiasm for science. And so he sort of grabbed that beast by the throat. And the next two plus years have in fact just been a complete trajectory upwards. He managed within relatively short order, probably over the space of a year, to generate not 1, not 2, but 3 very complicated, important human knockout cell lines. And then he spent the last year and a half characterizing them, and they have all of these wonderful, fantastic phenotypes. That work he has written up. We haven't submitted it yet, but it is going to make a very large splash when it comes to see the light today. He has a very lovely story to tell, and I think it's a great credit to his perseverance throughout this whole thing that he's managed to be standing here today and going to be able to tell you such a nice story. I think a lot of lesser graduates who really would have failed earlier on have not done that. Much to his credit. So that's it. I have to tell you that this is done in partial fulfillment of the requirements for his degree of Doctor of Philosophy. I would also note that he's titled his talk as kind of a metaphor of the 3 Musketeers. Okay, I would like to have hoped that throughout this 6.5-year sort of sojourn that I played D'Artagnan most of the time.
No.
Unfortunately, I think if you asked Ron honestly, he would say no, he was Cardinal Richelieu. So, in spite of that, I do think that we have managed to sort of keep ourselves together. We're both on the same page here. I'm very, very happy for you, Han, and I'm very happy for the story he has played today. If things should go well over the next couple of hours, at 4:30, in the 6th floor lunchroom, there will be a small, rather large celebration.
And at 6 o'clock, I'll be a stubborn herb. So. But thank you very much, Eric. Thank you for the kind introduction and the nice words. Yes, it has been a roller coaster. That's the animation you guys saw. It's been up and down, and in the end, it's a fun ride. So, so today, I'm going to talk to you about 3 nucleases, and I titled it All for One, One for All. The nuclease trio guards the cell's kingdom. So it's a story about the 3 major nucleases, CTIP, EXO1, and DNA2. What sort of roles they play in human cells. Oh, Mike, can you guys can hear me?
I can hear you.
We can hear you.
Sure? Ah! Just project. That's what Eric would say. But okay, so it's my intro slides. So I want to introduce you to my human model. His name is Generic. So Generic, if you decellularize Generic, you would get about 37 trillion cells. And in each of these cells, it hosts the human genome, which is about 3 billion base pairs in size, and they're nicely tucked away into 23 chromosomes, and they encode about 20,000 genes. So it's pretty amazing how 37 trillion cells that can just be a blob somewhere, it actually looks like, keeps us alive, they interact, and they coordinate each other. It could look like him, it could look like her, and, you know, keeping us alive. Now, as unimaginable, unfathomable this is, it is also ironic and very tragic that all it takes is as little as 1 mutation in 1 little cell to take a life away. And we know that know that too well. It's called cancer. So now, on a daily basis, our cells are subjected to sunlight, UV rays, poisons, and some we have metabolic byproducts that can damage our DNA or the human genome. So these things can cause single-stranded DNA breaks, interstrand crosslinks, bulky adducts, and DNA double-stranded breaks. And all these damage to DNA can cause mutations. And when a cell hears the word mutation, it just hates it. So unfortunately, our cells have multiple DNA repair pathways to prevent mutations when they come across damages. So DNA double-stranded breaks are probably the worst offenders. They pose the greatest threat to the genome because they can cause apoptosis, and they're very difficult to repair. And if they're misrepaired, they can cause mutations. So so when you have like if you get irradiated or something, it's going to produce reactive oxidative species, and they're just going to be randomly attacking your DNA, and they will create breaks randomly on both strands of the DNA on the sugar phosphate backbone. So you'll end up with single-stranded breaks randomly around their DNA. So if it's close enough, it'll just snap, and you'll get a double-stranded break. So basically, you have two open ends of DNA floating around. And so radiation isn't only the source of double-stranded breaks. DNA replication is the source of unwanted double-stranded breaks. So you have your nice fork traveling, and if it encounters a lesion, it will stall, and it has to repair and restart the fork. Now, if it stalls for too long, what can happen? Nucleases can just snip it, and you end up with a double-stranded break. As horrible as that sounds, fortunately, we have at least 3 DNA double-stranded break repair pathways. So in human cells, the main pathway is the classical non-homologous end joining pathway, and it is Ku-dependent. Eric's favorite gene is big donut, and basically what it does, it just rams together 2 ends of broken DNA. You get your repaired DNA. Now the other repair pathway is the homologous recombination pathway. It relies on it uses a sister chromatid as a template for repair. So it's a bit complicated. It takes longer time than the classical NHEJ. And this is the predominant pathway. Now, there's another little pathway called alternative NHEJ. It's basically the same. It works similarly to CNHEJ, except that it edits your broken ends a little more and fixes the DNA with microhomology. So a little bit more editing than CNHEJ. So the nucleases mainly play a role in homologous recombination, and basically my talk would be revolving around this repair pathway. So I'm going to run you through a bunch of slides to help you understand my story today. So homologous recombination, sorry, this is not homologous. In the NHEJ pathway, once it sees a double-strand break, the Ku70, Ku80 heterodimer latches onto the broken ends of the DNA and recruits DNA PKCS, which forms the synapse. And then it recruits Artemis to clean up the ends. Artemis is a nuclease. And then it is ligated by the XLF, XRCC4, and ligase 4 complex, and your DNA is fixed. Now, the problem with NHEJ is that it can be error-prone because of the editing that goes in here from Artemis. It can resect a little bit, and you can lose some genetic information. So it's a bit mutagenic. Now, HR, homologous recombination, is very different. It's more complicated. It is an error-free repair because it uses an undamaged sister chromatid as a repair for template. And it is important for repair that arises from a replication stress. And it is also required for restarting replication, stalled replication forks. Now, in humans, in humans and mammals, same thing. HR is upregulated in the S and G2 phases. So just to run you through this pathway really quick. You see a double-stranded break, the MRN complex gets in there. Then resection happens to generate 3' overhangs, which is then coded by RPA. Now next, with the aid of BRCA2, Raf51 is loaded onto the single strand to form a nuclear filament, and it bumps out RPA. And then you have your D-loop formation, which is aided by Raf54. It does the homology search in the other sister chromatid in red here, and then DNA synthesis occurs. And then it goes to, then the holiday junction is formed when Raf52 captures the captures these ends to anneal to the original sister chromatid in black. So then you form your holiday junction, it gets resolved, and you have your repaired DNA. Basically, nucleases will cleave these guys or resolve it, or and yeah, you have your fixed DNA. Now, resection has been. Can someone close the door up there, please? Thank you. So resection has been a black hole for a while. Nobody really knew who the players were, which genes, and really how this occurs. So it wasn't until 5 or 6 years ago, some elegant yeast studies really shed light on resection. So they found that at the double-stranded breaks, mRNA and SAY2 in yeast, or CTIP, gets in there with mRNA, joins into the action. It does minimal resection in there, just does some clipping a little bit to sort of unveil tiny little bits of single-stranded DNA, and it gets coded by RPA. And then this becomes a substrate for DNA2 and exon, and both of them work together with the Bloom helicase to do the long-range resection. So why is resection important? It's an evolutionary conserve process, and it's linked to checkpoint activation. It is critical for survival. It is important for genomic stability. Failure to regulate this process can result in human diseases. And one recently described a CTIP patient mutation. Basically, the patients suffer from microcephaly and several other developmental defects, really showing you how important resection is in development. And also, we know that current cancer therapies, chemotherapeutic strategies really target DNA replication machinery to cause double-stranded breaks in rapidly dividing cells. Now, and since it needs to be repaired by HR, nucleases could potentially serve as a novel drug target. So, and then now I'm going to talk to you about CTIP, exon, and DNA2 really quickly, and what we currently know about these genes. CTIP is essential in mice. It's not essential in yeast. And recently described CTIP functions is that, again, it interacts with the mRNA complex at double-strand breaks to promote resection and homologous recombination. It's required for proper checkpoint activation, and we saw that there was a patient mutation out there. And they are upregulated in several cancer cell lines as well. It's a busy protein. It seems to be a hub. It just doesn't just do resection. There's PCNA. There's a PIP box here where it binds to PCNA, hinting at its role in DNA replication, which has not really been shown. There's ATM phosphorylation sites, BRCA1 binding site, and it's a retinoblastoma binding site. Now, exon has been studied quite a bit. It is a 5' to 3' exonuclease. You just choose DNA all the way. Interacts with SGS1 in yeast or Bloom in humans for DNA resection. It is not essential for mice, but knockout mice are susceptible to cancer and they're infertile. Not essential in yeast, and they have been long known to be involved in the mismatch repair pathway and also homologous recombination. Now, the most recent described function in exon is that it also maintains telomeres by creating single-stranded DNA to create the T loop at the ends of chromosomes. So it's a really busy protein. Now, DNA2 is the least studied of all the three guys here. It is an endonuclease, so it makes clips off DNA. Again, it interacts with Bloom, and it is essential in yeast and mice. Important for homologous recombination, and it has been established as a Okazaki fragment processing. Plays a role in Okazaki fragment processing with FEN1. And in humans, they were found to reside in the nucleus and mitochondria, and also with some implications in telomere maintenance. And one of the latest things in DNA replication, its role in DNA replication is that it prevents stalled forks from catastrophic reversal in POMB, in yeast. Okay, so I just want to quickly summarize what we know. So CTIP is endonuclease. We don't really know that if that's true in humans. More work needs to be done. DNA2 is an endonuclease for sure. XO1 is the exonuclease, as its name suggests. And these two guys have roles in telomere maintenance. All of them are important for double-stranded break repair, homologous recombination. Now in yeast, DNA2 is essential, but not CTIP and XO1. In mouse, everything else is essential except XO1. And we really have no clue if all these genes are essential in humans. So just to wrap it up right here, for what we really need to know, the CTIP interacts with mRNA complex to initiate double-stranded break resection. DNA2 and XO1 then comes in to do the really processive and extensive resection that just reaches out KB lengths. So no CTIP, XO1 human, DNA2 human cell line available on the planet Earth, so we're jumping on this. So the studies that we've seen here, what we know about CTIP, XO1, and DNA2, the human CTIP, XO1, DNA2, they've been studied with this very, very common approach. Start with the siRNA-mediated knockdown. They throw in a bunch of DNA damaging agents, but mostly now, what we've seen mostly is just to cause double-stranded breaks, or lasers, and then they measure the response to the cell's response to repair the damaged DNA. So don't get me wrong, we have learned a lot. We also have learned a lot quickly. Now the problem I have with these studies is that, well, siRNA is never 100%, and we never know if these genes are essential or not once you knock it down. DNA damaging agents, you know, that's not what a cell really does. So it's kind of artificial, and that can mask the real functions of these genes in an otherwise unperturbed cell. So so we decided to create genetic models. That's probably the best way to study gene function. So we use the HCT116 human colon carcinoma cell line. It is a very easy cell line to work with. Its genome is very pliable. You can just knock stuff out happily, and it is the p53 pathway checkpoints are all intact as far as we know, and it's got 46 chromosomes, and the karyotype is relatively normal with tiny little bits of translocations, but nothing too crazy. So I've decided to make CTIP knockouts, as Eric said, XO1 and DNA2 knockout cell lines with the aid of RAAV-mediated gene targeting, and we've been using this for many years. So I decided to go with conditional knockouts instead of just straight knockouts because, you know, given how essential they are in mice and yeast. So I created conditional knockouts for CTIP and XO. Now these two cell lines, I was able to derive them in parallel, same timing. I knocked out the exon 2, I flocked the exon 2 in CTIP and exon 3 in XO1, and once you take it out, it creates a frame shift mutation and protein's dead. So, and DNA2 took me a little longer, because after I knocked out 2 alleles, the third allele, we realized that it was triploid. So it took forever. Oh my God. So with Beth came in, and she helped me out a little bit with screening these cell lines, and finally I got the cell line, and I was such a happy boy. So basically the story here is that I have a lot of data on CTIP and XO1, and DNA2 definitely needs some catching up to do. All right, so next thing I did was to make my life a little bit easier. So I created a Cree-ER system, an inducible Cree-ER inducible system. which is a Cree that's attached to estrogen receptor. So I made stable cell lines, stable Cree ER expressors of these mutants. And without tamoxifen, they're going to sit outside of the nucleus in the cytoplasm. Now, when you hit it with tamoxifen, it's going to bind to Cree ER, and it gets that signal, and it's going to get into the nucleus, and then it's going to recombine the exon out, and you'll get a null cell line. Very easy, very effective, very inducible. Now, first thing I want to show you, now we're getting to the juicy parts here. Cree ER is functional. So up here is CTIP, and down here is XO1. I harvested cells from day 1, 3, 5, and 7. And this is going to be a very common theme in the rest of the slides. I harvested cells at day 1, day 3, day 5, and 7s. And so we have wild-type cells over here. In the ethanol control, CTIP is still there. I added tamoxifen into my wild-type cells. They're still there. Now, my CTIP hets, well, in the controls, yes, they are more than 50% down from their wild-type counterparts. And then as soon as I hit them with tamoxifen, bam, it goes away. Same thing with XO1. They're here, goes down a little bit. I don't know why this popped. Then I hit it with tamoxifen, it is gone. So that's the first thing I showed. Cree ER is functional. So I then wanted to see if they're alive or dead without these genes. So I did an MTS colorimetric assay, which basically measures their, it's a colorimetric assay that measures their NADH levels to see if they're alive. So my controls with ethanol, in black line, that's a wild type. Blue is CTIP. They're growing fine. XO1, and if you look at the cells, they're just they're struggling a little bit. And they grow, the slowest growing cells of all the three here. Now, if I add tamoxifen, it just flatlines. There's no growth. They're just sitting down on a plate. They're not doing anything. They're dying. They're dying. So CTIP and XO1 are essential genes.
Did you measure death?
No, didn't measure death. It's not proliferating. It's dying. Could do some tunnel assays or something like that. So when these cells in tamoxifen were just sitting down, they looked really weird. They looked ugly. They're really huge. They were binucleated. So I told Eric, please come take a look. What the hell is this? Oh, they look like they irradiated. Did you irradiate them? So nope, I didn't do anything to them. So, well, it looks like they've got a lot of DNA damage. So, well. So the first thing I did was to strip this blot and reprobe for P53, which is a marker for DNA damage. And turns out they are suffering. They are just having quite a lot of DNA damage, and so that got things really interesting. So same thing with XO1. Once you hit it with tamoxifen, P53 levels go up, and although the hets they have a little issues, but not as high as when they're gone. And the surprise, this is quite mind-boggling, is that we didn't treat them with anything. No DNA damaging agents, nothing. So with this amount of damage that I've seen, I've decided to analyze the karyotypes. So I took these guys and sent them to our cytogenomic shared resource, and Leanne Oseth did analysis, and she took all these beautiful pictures. And so I have 3 time points. Basically, I'm trying to find, one is at CTIP is at day 7 and day 5. I didn't make a mistake. Your eyes are not lying to you. I was trying to stretch out, trying to find the latest time point where we could get metaphase spreads. And CTIP went all the way to day 7, but XO1 by day 5 was the last time point I could get any metaphases. So from day 0, they look relatively normal. Got little arrows here if you can't see. They're breaks. And then at day 1 in CTIP cells, they start getting fragmenting and just a lot of aberration, just a lot of abnormal chromosomes. And by day 7, it gets worse. Same thing with XO1. You get a lot of breaks, radials, And by day 5, it's a mess. So I have blown up pictures for you. So CTIP at day 0, we have 1 or 2 breaks. That's pretty normal. When you hit day 1, you get radials. Now radials are pretty severe phenotypes, and more radials here. These are breaks and fragments, and another break here. So Leanne can see it. And by day 7, same thing. It just gets worse. You get more radials, radials, radials. You get fragments, little pieces here and little pieces here and there. So it was crazy. It was just ugly. Exo-1, same thing. Day 0, day 1, start seeing radials, radials. Looks like a chicken foot. And just a bunch, it's just a huge mess. And by the 5th day, ew, right? More, you still see radials, fragments, and aneuploidy. So we had there might be even some complex exchanges here. I mean, this looks like pigtails. It's my favorite. It was very difficult to quantitate. But so Leanne and I sat down and we said, okay, well, let's find a reasonable way to score these phenotypes. So we came up with a table, and basically try to summarize everything and see what happened. So, so the values are in percentage of the total metaphases analyzed. So as far as breaks go, it's all time dependent, and once you add tamoxifen, so the CTIP analysis, they have more breaks. Same thing with Exo-1, you get more breaks. So these are metaphases with between 4 and 15 breaks, or more than 15 breaks. So they keep climbing, and same thing with fragments. You see more fragments in a time-dependent manner. And also we looked at chromosome counts. They also go, it goes higher. We see tetraploid cells in CTIP, and but interesting, we also found some octoploid cells, so that's 16N. It's pretty dramatic. In Exo-1 cells, the zeros here, we're not seeing much aneuploidy because there's so much fragmentation going on, and we just decided, it looks like it's 46 chromosomes. Let's just sort of leave it as there. And but at the last time point, you start seeing tetraploid cells. Now, radials is a pretty severe phenotype. They're mainly they're seen in Fanconi anemia patients, really sensitive to interstrand crosslinks, blinking agents such as cisplatin. And so we see an increase in radials also. Over here, we see, compared to controls, we see 16% and 13% comparable. And then in Exo-1, you see a lot more of your, this frequency of radials showing up. So, trying to, I don't know anyone has done this before, but I just wanted to have a good bird's eye view of what's going on, how these chromosomes are being fragmented so much. So I had some time and I just, tried something cute. I transfected these cells with H2B EGFP and took movies. So, oh yeah, I gotta use my mouse. What happened? Why are you doing this to me? Mike, help!
He's gone. He didn't pay with us the first time.
Oh, shoot. Sorry, guys. Yeah, I'm trying to do it. What is going on here? This is wonderful, guys. Back, please. Come back to me. Okay. So I have a wild-type cell. It's oh. Okay. Forget a movie. I'm forgetting about a movie. Yeah, it's just gross. Anyway, I see a bunch of micronuclei and
And heads, there's lots of heads.
and lots of heads. That's right, lots of heads. So yeah, so you can see these. I don't know if you, just really quick, you can see some little chromosomes here, and then they're just fragments. Just boom, there's lots of micronuclei. They're not aligned nicely. It's just ugly. But anyway, they suffer from mitotic errors and a lot of micronuclei, really indicating incorrectly repaired or not repaired damaged DNA. So from here, I'm just going to summarize really quickly. The CTIP and XO1 floxybromide pre-ER cell lines, they're working. It's functional. Cells cannot survive without CTIP XO1, so they're essential in human cells. And once CTIP and XO1 is absent, you see a ton of endogenous DNA damage. So from our karyotype analysis, also we noticed that, oh my God, genomic instability. It's just ugly chromosomes that we've all seen. So and again, just to remind you that they were not exposed to any DNA damaging agents, nothing. So it's a bit shocking to see how much damage can go on without these two genes. So what is next is my PowerPoint slide. Okay, so taking note that there are mitotic errors and aneuploidy. So I wanted to take a look at the cell cycle phase. So I did an experiment. It's called an EDU incorporation assay. It's similar to VRDU, except it's this EDU. It's very simple. I pulse the cells with EDU. It's going to incorporate it. Cells are in S phase. They're going to incorporate the EDU. I remove the cytoplasm, stain it with propylium iodide for DNA content and another color for EDU, and then run them through FACS. And this protocol was given to me by Spencer. It's a very good protocol from Shima Lab. So what I have here is a pretty busy panel. So on your x-axis is propylium iodide. It's collected in a linear gate, and that is the color for the EDU. So if you look at this here, there are three populations you want to identify. So it's G1. There's your S phase that's incorporating EDU over here, and your G2 phase, where it's not incorporating EDU, and it has twice the amount of DNA that you started with. So on your left side here are controls. You have wild-type, CTIP, and XO1 mutants. And over here, I added everything with tamoxifen. And again, it's days 1, 3, 5, and 7. So wild-type cells, fine. You're just looking fine and happy. You have all your G1, S, and G2 populations sitting nicely. On CTIP, first day doesn't look very much anything, not too different from wild-type or the controls. Now, as soon as it gets to the second day, I start noticing that their G1 and G2 phases disappear, and they shift up, indicating that DNA replication is going on. So that's really odd. And then the DNA replication is continuous. So there's continuous DNA synthesis that they keep expanding this. They keep accumulating with more than 4N DNA content, and they get out of the linear range. So it's a bit crazy. It's mind-blowing. I couldn't explain this at first. And so now same thing with the XO1 cells, except that it happens faster. And same thing, you see a shift in the G1 and G2 populations. That they're incorporating more EDU, and they just get out of your linear range. So this was a bit crazy, and I just couldn't understand what was going on. But all I know is that there's increased DNA replication initiation, and the synthesis is continuous, and they just don't stop, and they accumulate ridiculous amounts of DNA. So trying to resolve this population, I collected my data in log scale, and this is just for this is forward scatter, and just collected in log scale. And again, you see 2N DNA content, 4N DNA content. And once you throw in tamoxifen, the population starts shifting over to the 4N. And by day 5, you start seeing a lot, pretty much most of your cells are in getting out of the 4N gate here, like here. That's tetraploid, it's 8N. And then by day 7, in the CTIP cells, you see some octoploid cells, and not so much in the exon mutants. So this really correlates with what I've seen in the karyotype analysis here.
Did you also look at sub 2N? So it looks like you've got even micronuclei. Little stuff up here too.
That's true. There's a lot of micronuclei. Yeah, yeah. So that's what I've tried to show the mitotic movies. And there were more traditional cell cycle FACS plot looks. Again, this is in linear range and does not look like endoreduplication. It is just a mess. As you know, so your G1 peak, your S, and your G2. But I just want to show this, just show you guys that you guys are familiar with. So from this assay, what I saw is increased initiation, there's continuous DNA synthesis, and aneuploidy. CTIP reaches to octoploid, exo-1 tetraploid. So at this point, you're questioning, it's like, what is the source of DNA damage? And really, right now, it hints at DNA replication. So that's probably the only way where that damage could occur without the presence of DNA damaging agents. So I took a look at the replication track lengths of these cells, and I'm very, very thankful. I'm grateful to Tsuyoshi from Shima Lab and also the Sobak Lab for helping me out with this complicated assay. Oh my God, it's a pain in the ass. So we have, what I did was I incubated the cells with deoxygenin DUTP 15 minutes, and then followed by a second label, EDU. And I labeled deoxygenin DUTP in red and EDU in green. So if you have wild-type controls here, and these are median lengths that represent these green tracks here after an origin is fired. So in your wild-type cells, it's about 15, and then 15 micrometers. And in CTIP flox cells, it's about 14.4 and 11, much shorter in the exo-1 hets. But once you throw in tamoxifen, it is drastic. It gets shorter and shorter. Same here in exo-1, it gets much shorter, and it gets even worse here. So I quantify these guys. I came up with some box plots here. So these are wild types. These are control CTIP and control exo-1 cells. And then I have CTIP at day 1 and day 3, exo-1 and tamoxifen at day 1 and day 3. So it's pretty drastic. You start from a relatively normal length fork. It just drops down all the way down. So really indicating that in the absence of CTIP and exo-1, it's really affecting DNA replication. But and then I thought, what is going on? Is there really something going on with these with the replication fork? So the next thing I looked at was I looked at chromatin pCNA. So modified pCNA would indicate that the forks are encountering tons of damage and they're just stalling there. So over here is my wild-type lane, and the controls are here for CTIP and exo-1. Once I hit them with tamoxifen, you see a gradual shift of modified pCNA, and this looks like a monoubiquitinated pCNA, and really this piece of data here reaffirms that DNA replication is suffering without the presence of CTIP and exo-1. So the next thing I wanted to do was to check for stalled replication forks. And I did that with RPA staining. And show you here again, you have a ton of foci once you add tamoxifen right here. And it goes, there's a big spike, and it goes down here in the CTIP. I graphed them here. There's about 500, I scored 500 nuclei. In XO1, you also see a similar pattern. You see a big jump, and it drastically goes down, though. The downward trend isn't as steep as what we see in CTIP cells. And these are my controls with hydroxyurea. So the forks are stalled. And then I looked at FANG-D2 foci, which is also a very strong marker for stalled replication forks. And I see the same trend where I see a big spike in the first day after adding tamoxifen, and then it goes down. And same thing with the XO1 cells. So the phenotypes are very identical between CTIP and XO1. Then I looked for double-stranded breaks. So I stained them with gamma H2AX. So these cells are screwed up, man, I tell you. So they have the gamma H2AX foci going up the wazoo, and this is the wild-type cells treated with camptothecin, which causes DNA double-stranded breaks. And you see that basically the pattern is that without CTIP or XO1, the levels of H2AX foci are high, and they just persist over the course of the experiment. Now, one more thing I wanted to look at was 53BP1. This is a bit off, but 53BP1 is a very hot gene right now. It is a marker for double-stranded breaks, and it also is a marker that double-stranded breaks are being repaired by NHEJ. It is a very strong promoter for NHEJ repair. So what I see here, same thing. I see in CTIP null cells, they go up, and they stay there, and they persist. XO1, you see a bump at first, but again, they still persist throughout the course of the experiment. So seeing how the patterns of the foci going up, going down, or going down, going up, and persisting there, I kind of wanted to know, where are these proteins going? So I took chromatin fractions and also whole cell extracts just to see what are they being degraded? Are they just dispersing from nuclear foci? So I took these days 1, 3, and 5 instead. Just took out the 7s, too many time points there. And I had plotted for CTIP and XO1 just to show that they are gone as my controls. And so these are the top panel here are from chromatin fractions, and the lower panel from whole cell extracts. And they don't necessarily reflect each other's levels. So in the chromatin, FANG-D2, we see a spike in day 1, and it goes down. Same thing with XO1. And when I looked at the chromatin fractions, again, there's a spike, and then it goes down. Same thing with XO1. There's a spike here, and it goes down compared to the wild-type counterparts, the controls. Oh, sorry. And in the whole cell extracts, this is they're gone. They're gone. They're just being degraded. So this is the first time I've seen this. It was quite surprising. And then we looked at RPA. RPA was the same thing. It seems like they're degraded as well. So if you look at the chromatin fraction, it gets lower and lower and lower over time, but not as severe as XO1. It is completely off the chromatin. And the whole cell extracts, so there's a ton of RPA, and a little bit of a change in intensity would really indicate that they're getting degraded. And then 53BP1 is the same thing. This you see an upward. You see them building up, and you see the same thing here in CTIP. And then in XO1 cells, see pretty much the same thing. They're persistent there, and the whole cell extracts they're not getting degraded, but they are in CTIP. It's kind of weird. So, a quick summary here. I'm trying to piece all these little pieces of data together. The gamma H2AX and 53BP1 foci really tell me that the double-stranded breaks are occurring at alarming levels, and NHGJ is actively joining them back together. So and there's a paper that really reaffirms my conclusions, is that 53BP1 mediates productive and mutagenic DNA repair. So this paper showed that 53BP1 is responsible for all those radials and fusions that we've seen, just exactly the same that what I've seen in my karyotype analysis. Just like these guys. 53BP1 has been shown to be responsible for these guys. Sorry. Now, the other thing was my fiber assays and my DNA replication assays. The fiber assays is show that DNA replication is severely disrupted, and also in conjunction with FANG-D2 and RPA foci indicates that the replication forks are stalling. So at this point, I'm just trying to figure out why they're stalling. I'm trying to find an explanation of why are these forks stalling, and there's no DNA damaging agents. There are no replication inhibitors, nothing so far, and everything occurring here is endogenous. So it turns out there's a good explanation for this, and there's plenty of them. So it turns out the human genome is it's a landmine for DNA replication. So there are unstable inverted repeats, the repetitive regions that can cause replication fork stalling, and there are chromosomal fragile sites. There's about 100 of those identified, and even more recently, there's another class of fragile sites, early replicating fragile sites that can break, they stall and break really quickly, and these are all located in repetitive regions such as line elements, sign elements, telomeres. So telomeres and all, it's just causing a lot of problems in the absence of CTIP and XO1. So the next thing I found, you might have caught this earlier, that CTIP and XO1 are co-regulated. So this has never been shown before, and it's a bit surprising to us as well. So in the CTIP null cells, when I add tamoxifen, and they go away, but surprisingly, XO1 also goes away. And this is the same thing when I add tamoxifen to my XO1 mutants. XO1 goes away, and CTIP is also downregulated, and it goes away. Together with that, BRCA2 also goes away in both these cell lines when XO1 is, when CTIP is in CTIP null cells. Same thing with XO1. BRCA2 is degraded. FANG-D2, like we've seen earlier in the immunostaining, they are gone, and so is this. And RAD51 is also gone. So one more thing was really shocking was CHEK1 is also disappearing in these two cell lines. So this indicates that CTIP and XO1 coordinately regulate FANG-D2, BRCA2, RAD51, and even CHEK1, and that's never been shown before, and this is quite difficult to explain. So the next thing we try to figure out, how are these proteins downregulated? Is it by proteasome, or is it by the proteasome? So I treated these cells with MG132, which inhibits the proteasome. So these are my controls here. In wild-type cells, I added MG32 24 hours after adding tamoxifen. 12 and 24 hours after adding tamoxifen, I harvested these cells. And in my, so they're all there in my wild-type cells. But as soon as I hit them with tamoxifen in CTIP cells, you see that FANG-D2 goes away, like we've seen over here. CHEK1 goes away, and RAD51 goes away. And that's the same thing for XO1. They're going away without MG132. And once I throw in MG132, you can rescue the levels. So this shows me that the HR factors and check1 are being degraded by the proteasome. So this is a bit crazy. Check1 is disappearing, but there is an explanation for this. So several years ago, many years ago, 2005, check1 degrades under chronic genotoxic stress. So that pathway is activated. The ATR check1 pathway, when it's activated for long periods of time, check1 will get. You see check1 getting degraded. I mean, that's a necessary step for check1 to get degraded, so we can restart the cell cycle again. So moving on, I wanted to show that check2 is still present. So the other checkpoint is still functional, because check1 is gone, and it is quite crazy what's going on. And I just want to show you that check2 is still available, it's still right there, and it's upregulated in the absence of CTIP or XO1. And it is activated. I use an antibody against phospho-check2 at threonine 68. Right here and right here, it's upregulated. ATM is there, and ATM is also activated. So this is a strong marker that double-stranded breaks are occurring, and the ATM check2 pathway is activated. And I probed for Ku86 to really to show that NHJ is still NHJ is present, and it's still there. There. Now, this is the last piece of the puzzle right here. DNA2, like I said, this is the last outline I got, and I wanted to see how DNA2 would interact with CTIP and XO1. So, we predicted that it would be the same. It would go away similarly like XO1 and DNA2, because there are three nucleases that are working together so closely. So again, in my CTIP null cells, as soon as I added tamoxifen, I think I messed this up. No, I didn't. Sorry, these are my controls. So if I plot it with XO1 right here, in my CTIP cells, they're gone away, and DNA2 is gone as well. And then when I plot for, I think my plot is, no. In XO1 null cells, treated tamoxifen, it's gone, and DNA2 is also gone. So really what I'm trying to tell you is that that these DNA2 and CTIP and XO1 are all, they are co-regulated. And finally, I also want to show you that in DNA2 null cell lines, they show the same phenotypes, where BRCA2 over here, FANG-D2, RAD51, and CHEK1, they're all degraded. So FANG-D2, oh my goodness, I'm sorry. They're all co-regulated, and they coordinately regulate the HR factors. And just to show you that this is not cell line specific, I collaborated with Jung Eun from the Sobek lab, and she knocked down CTIP, XO1, and DNA2, or DNA2, in the GM637 cell line, and that's more normal human fibroblast cell line. And what happens here is, when she knocked down CTIP, here's CTIP, she also sees XO1 going away. When she knocks down XO1, XO1 is not here, CTIP also goes away. It's very similar. When she knocks down DNA2, it's gone, CTIP also goes away. And that's the same thing. When she knocks down CTIP, DNA2 goes away. Knocks down XO1, DNA2 goes away. It's downregulated. Same thing right here. But yeah, that's gone. And finally, one more plot showing that FANG-D2 also gets degraded in the absence of CTIP, XO1, or DNA2. It's downregulated. So really, DNA2 is also co-regulated with CTIP and exon 1. And we see the coordinated regulation of these 3 nucleases of FANG-D2. And again, this is not cell line specific, and it applies to all, I'm guessing, all human cells. So anyway, I tried to put together this model, my super model. So here's my replication fork. We have your leading strand right here. So with the fork stalling, I showed that there were replication tracks really short. There's pCNA modification. There are FANG-D2 RPA and foci. Is that when the fork runs into a repetitive region, it can form. You can see G quadruplexes or some really difficult templates where the polymerase cannot go through. So what happens is it's going to stall. You get full of RPA. Or you can even see weird structures like these, hairpins, whatnot. pCNA gets modified. Your ATR and check1 pathway gets activated. Now, FANG-D2 gets there, and RAF51 and BRCA2 also gets there, as we've seen from the foci. The FANG-D2 foci over here. Now, keep in mind that these are cells that do not have CTIP, exon 1, or DNA2, as we've seen from the co-regulation. So when they're not around, RAF51 and BRCA2, they can't really load on RAF51 onto your single-stranded DNA. So what happens is that it's stalled, nothing's going on, it can't restart, it can't repair it. So the cell says, hey, I need to move on, so it degrades all these guys. And at the meantime, as check1 gets activated, the damage is still there. So check1 just keeps getting on degraded and degraded. There's no time for it to build back. So that's and all of this is responsible by the proteasome. These 2 proteins get degraded, and what happens is your fork here collapses into a double-stranded break. And from what we've seen, BRCA2 and RAF51 is degraded, so HR is really not there anymore. And with the with 53 BP1 foci going up, skyrocketing so high, there's only one more explanation, is that NHGJ is doing the mutagenic repair that results in all these ridiculous chromosomes. So, so the take-home message here is that CTIP exon 1 are essential in human cells. They play essential roles in DNA replication. And the DNA replication landscape of in the human genome is just filled with landmines, repetitive regions that can cause a lot of stalling. These 2 genes also regulate HR in a very, very important manner. And I've also found novel interactions between CTIP, exon, and DNA2, that they are co-regulated. And they also coordinately regulate FANG-D2, BRCA2, RAF51, and check1. So future directions for this, for my work, submit the manuscript. And to finish up the DNA2 characterization, complementation of these guys for more structure function studies. And then I think the really bread and butter experiment for the next, to continue the study, is to do SMART, or single molecule analysis of replicating DNA of chromosomal fragile sites or early replicating fragile sites where all the repetitive regions are. So you can actually look at what kind of how DNA replication is altered in that region. So my previous work and ongoing work, what I have, I've published this paper, like Eric has mentioned, and just a few days ago, Sobek Lab and our lab published this paper in Human Molecular Genetics, where they use micelle line CTIP mediates replication for recovery in FANG-D2 regulated manner. It's very exciting. And also, this is another piece of work, another paper that is going to be submitted. It's in a collaboration with Judith Campbell and Caltech. And that's all I have. And I want to acknowledge Eric, the poobah, calls himself. My committee members, I am very glad. I couldn't have picked a better committee. It's been very supportive. The Hendrickson lab members, Beth, I don't know how she does it. She's a mother of 3. and she has released 2 cell lines within a year and a half. Adam, Yinan, Ben, and these are previous lab members that work closely together. And neighboring labs had a great time with the Sobek lab, working with Alex and Zhang. A lot of discussions going on, and I think when I leave, they're going to get me a baby monitor, they say, so they can hear me in the other lab. Blinsky lab, thank you so much for reagents and having great conversations with Bob, Iman, Jordan, Ryan, Tenji, and Shahan. Shima lab, Tsuyoshi, and Spencer really had all these complicated experiments done and kind of walked me through the protocols. And Harris lab, there's just too many of you. The Duncan Clark, I don't know if he's here, but I'm I'm eternally grateful, really, for him. He let me use his confocal microscope and the Delta Vision diode. Leanne, she was so patient with me and with analyzing all the karyotypes. The Flow Cytometry Resource Center. The department, the biochemistry department, and thank you all, the PIs, for working so hard to really, I think it's not a trivial thing to keep this graduate program going. And I'm a foreign student. I'm very, very thankful. I'm grateful for the generosity of everyone. You guys are paying me, basically. I have some pictures. You know, my grandpa is 93. I go home and visit him. I miss the food. And my husband, John, thank you so much. He put up with so much. Oh, my God. And stupid thing. That's my sister. 2013 was a big year for us. She got married, I got married, but I still think I look better in that dress. My family tucked behind there. I'm sorry, guys. Lab, we had skiing. I mean, Eric couldn't have picked a better day. That day was just powder. It's the best. Classmates hidden behind my grandfather. Oh, that's it. That's all I have, you guys. So thank you for coming.
We've run over more than 10. Why don't we take a few questions?
Sorry, guys.
Yeah, so you talked about the whole regulation.
Yes.
When you see regulation, are you saying transcriptionally? Because it looks like proteins are being degraded to longer complex. When you say regulation, I guess I'm wondering what mechanism are you thinking is transcription?
Yeah, it's not transcription. I mean, I should have used cyclohexamide together with the MG132 to see if it's that. But no, I think what happens is when the forks stall, everything's just gonna they're trying to repair, but without the nucleases, they're just stuck around there and they can't load on the right places. And what happens is that the cell says, okay, you gotta get out, I need to repair this. So they clear it out with the proteasome, and then they make a double-stranded break, so NHEJ can come in and repair. And so that's what I meant by regulating. Yes, Jeff.
At the beginning, you summarized data from the mouse on whether these nucleases are essential or not.
Correct.
I can't remember the specific case, but there was at least one case where it's not essential. Is that right?
In mouse?
Mouse or
Yes, Excel one was not essential in mouse.
And were there any cases, are they all essentially used?
No, they're not essentially used.
They're not.
They're not essentially used.
So, in light of the massive problems that causing cells to be reconciled, what's your best idea of the difference?
So structurally, the gene isn't different at all in between mouse and human. But I know that in mouse, they've showed that it's very important telomere work and telomere maintenance. Now, we've published a paper several years ago. I mean, Eric with what's his name? Yang Bao, postdoc Yang Bao. So they showed that Ku86 is essential in humans, but it's not essential in mice. So that's the same discrepancy you see. And they've identified the problem is that it's telomeres. So without Cu86, the telomeres start fusing and they fall off, whatnot, they get shorter. And I'm trying to extend that the essential function could be telomeres in humans, but less in mouse.
What you're suggesting is that the replication problem in cells is primarily caused by repeat.
Yes.
Areas of the genome that are loaded with repeats.
Uh-huh.
You don't have very much of it.
Right.
So could that explain?
That could be it. I think yeast exo1 is, I don't remember how different it is. But the studies in yeast really are predicated among, very tightly in the sense of double-stranded breaks. So they have just make a double-stranded break, clean break with the HON nuclease system, and they just look at that. And I don't really. Sorry. Yes, I do know an answer for that. That in exo1 yeast, they have shown that it is involved in DNA replication restart, just like stress. But I don't think it's stalling as crazy as what's going on in human cells. That's just my guess, like what you said. Ah.
So you showed that when you disrupt these genes, basically they, in some instances, they can continue to replicate DNA, but they don't divide. So you end up with tetraploid cells.
Yep.
Is there a precedent for this?
Ye-
Can you tell me another gene where this has been seen before?
Another gene where this has been seen before. Not that I'm aware of. But I think what can happen is sometimes once you get to your G2M phase and there's that damage, that checkpoint is kind of leaky. So they can just go past that in very rare cases, I think. And then they'll get from G2 and then they just go into mitosis. And that's when you see all these ripping up happen, mitotic errors happening. So that pathway is kind of leaky.
Makes me think like there's a defect in cytokinesis.
Well, yeah. I mean, that's really out of my range right here. It's really far out right there. Cytokinesis and it's hard for me right now. I mean, I don't have the sort of the specialties in that field to sort of connect with this. Huh?
Yeah, go.
Yes. So, looks to me, PCNA is also disappearing, right?
I don't think it's disappearing. It's still in the.
It's here, but it's not.
It is on the chromatin. It's modified.
No, but it's going away. You're losing all the unmodified PCNA.
Okay.
You have very little left, and that's modified, and that's on chromatin. So
It's going away.
disintegrating.
Okay.
So, for different technologies, they need PCNA to be consistent.
Correct.
That's why maybe it's not.
Ah. So so I don't know if that's the case or is it the origins are dormant origins are firing to rescue the stalled forks, and they just can't. They fire a little bit and they just get stuck when they meet the other fork that's stalled. So that's my guess. That's why it's so short. But in terms of having it being processive or without PCNA, I don't really think that's the case. I think it's just origins firing, trying to rescue the stalled forks. That's why it's so short.
I'm sorry.
Okay, well,
I have answered.
Sorry. So you have normal replication going on.
Normal, long
Your
process.
DNA content keeps stuffing.
Yes.
What do you reckon? How could this possibly happen at the same time? And I don't know how you could even have an answer for this.
I think I might have an answer. So if all the regions I think it's sort of like a timing thing. So again, this is all a time course. So after I get rid of CTIP and exo-1, I mean, this whole big genome, it's replicating, it's going through. Eventually, all the forks are going to meet their demise, right, at the repetitive regions. They're just going to stall there. And then you have all your dormant origins firing, and that's more than normal. The whole genome's dormant origins are firing and firing and firing, and so you just keep incorporating EDU. It just won't stop.
Fibrosis in day 3.
Day 3. I don't think you'll see anything in day 5.
All right, well, it sounds like Vinny's going to have a lot more questions as well, too. But why don't we thank you, Han, for the presentation. Once again, hopefully we'll see you all at 4:30.
Okay.
I just emailed her.
HR?
She didn't get this one.
Okay. Thank you. I'll be